Okay, so photosynthesis in higher plants. So we already know what is photosynthesis. So what is photosynthesis? Photosynthesis is nothing but a physico-chemical process by which green plants use energy to synthesize organic compounds. So this process basically takes place, okay, in the plants, photosynthesis. So the basic, this one, the process which is there, like out of the catabolic and anabolic process, this comes under the anabolic process because there is a formation over here. Okay, so we all know the basic equation of the photosynthesis. In the presence of light, there is a sunlight, it gives you glucose, water, and oxygen. Okay, so this is the basis for the life of whatever is present on the earth. Okay, and because of this process, only fresh air we can breathe. Because what happens in the photosynthesis is oxygen is released. So whatever carbon dioxide we exhale, that has been taken up by the plants, and in turn they give us what? Oxygen. So this all takes place in the green parts of the plant with the help of the sunlight. Okay, so we are going to see what are the reactions which usually takes place during the photosynthesis and some of the experiments which are involved in it. So photosynthesis is very important because it's a primary source of food and health, and also it releases oxygen to the atmosphere. So now let's see certain experiments which were done earlier by the scientists. Okay, so the early discoveries. So the first discovery was done by Joseph Priestley, right? So Joseph Priestley. Now what did he do? Okay, he just put an experiment using a candle and a bell jar. And for the sake of a sample, he took a mouse. So here he is basically trying to show that air is very important in the growth of the green plants. Okay, so what he did was he kept this mouse in the closed space, that is that glass bell jar. Okay, so what happened was after some times to see that whether the mouse is going to die or not. So obviously what will happen if the oxygen is not supplied to the mouse, that living organism is going to die, right? So here what he did was in that particular glass jar, okay, he kept that mouse closed in that, okay, and it will die. But if instead of just keeping the mouse, if he puts a plant over there, any plant, okay, now here he used was the mint plant. Okay, so he kept it over there. So the mint plant is kept in the bell jar. So what will happen is not the mouse will die, and even if that candle also will not get extinguished. So what he concluded was what is the foul air which was initially produced by this mouse, that is that animal. It was converted into a pure air by the mint plant which was kept over here. So that was the experiment done by Joseph Priestley by using a mouse, okay, then a candle, then mint plant, and a bell jar. So here he is explaining the role of what air is important for plant growth. Okay, next experiment which was done by Julius V.S. Now this person did this experiment to show that these plants produce something which is called as glucose. okay, which is initially stored in the parts of the plant in the form of what? Starch. And in the photosynthesis, starch is only the first visible, what do you say, product that can be seen. Okay, so here the statement was what? Green parts of plant make glucose. And what happens? It is initially stored in the form of starch and stored in the form of starch. Third person is T.W. Eagleman. Now he used a prism. Okay, now as we know that when a light passes through the prism, we get 7 different colors, which is known as the Vibgyor, right? So here what he did was he used a prism so that the light could be split. Okay, to what do you say? Split the light into Vibgyor. Okay, so with this, he kept a suspension of what? Aerobic bacteria. Suspension of aerobic bacteria. Aerobic bacteria means what? Those bacteria which breathes in presence of air. So hence they are called as what? Aerobic bacteria. So when he placed the suspension of aerobic bacteria and when he illuminated that particular split light through the prism which produced the, what do you say, the Vibgyor, what did he found? He found that the bacteria which were accumulated, it was in blue and red light spectrum. Okay, so according to him, he says that different wavelength of light, okay, also has an effect on the photosynthesis. Okay, so effect of different wavelength of light on photosynthesis. Okay, in the same way, one more person, I think this is the fourth one, Cornelius Van Niel. He did an experiment. So it was basically, he used over here the purple and green sulfur bacteria to show that the photosynthesis is a light dependent process. Okay, so that was his study where he used the purple and green sulfur bacteria to show that photosynthesis is light dependent, means it needs light for the process to occur. Okay, so whatever he used over here, the purple or the what do you say, green sulfur bacteria for this experiment to show it's just a light dependent in the reaction. Okay, so this was given by this Cornelius Van Niel. Fine. Now, these are the experiments which were to confirm that what actually happens in the photosynthesis. Now, in order to know what are the reactions which takes place in photosynthesis, you should know that where it takes place. Means you should know the site of what? Photosynthesis. Okay, where does it actually happen? So, site of photosynthesis. Okay, now every green plant has something which is called as what? Chloroplast. Right, now what are these chloroplasts? Chloroplasts are nothing but green plastids. So this is where the photosynthesis actually takes place. Okay, so where is this chloroplast present? Inside the leaf. In the mesophyll cells. Okay, so even though a chloroplast looks simple, it has certain parts inside it, because chloroplast is generally what? A membrane system. So this is the actual compound which is responsible for trapping the sunlight, which you get from the sun, and in turn it will produce what? The energy molecules, or whatever you say, ATP or NADPH for the process. Okay, so usually this particular membranous system is going to consist of what? A grana, stroma lamellae, then the fluid stroma. Okay, so this particular membrane system, what it is going to do? It is going to trap or absorb, you can say, the light energy, so that it can give you ATP and NADP. Plus here what happens? Some of the enzymatic reactions also will take place in the stroma, which is nothing but the CO2. Okay, that is the sugar synthesis will take place over here. That is why we get a glucose as a product of the photosynthesis process. So here what happens is generally the light energy is absorbed by whom? The grana of the chloroplast, so that it can synthesize the ATP and NADPH. Now, this is applicable for what? A light reaction. In the same way, you have dark reaction as well. So, in the dark reaction, what will happen is whatever CO2 is there, that is going to get reduced to sugar. So, this is the dark reaction. So basic two types of reactions are there, okay, which usually happens in the chloroplast as a part of what? Photosynthesis. So this chloroplast is going to have, it's a membranous system. So it will have grana, okay, thylakoids, right? The stroma lamellae, which is the connections which it is having. And then the stroma, that is the fluid which is present in them, with an outer as well as inner membrane. Okay, so for this to happen, there are certain pigments which are involved in the photosynthesis. Okay, so it's like based upon the colors or a chromatogenic pigments, okay, which we are going to see. Okay, so the pigments involved in photosynthesis. So basic four pigments are there which are involved. Okay, so the first one is the chlorophyll A. Okay, then chlorophyll B, then xanthophyll, and last, carotenoids. Okay, so chlorophyll A and B. This basically it has a color which is bright blue green color. Whereas chlorophyll B is yellow green. So both are almost green, but a blue green and yellow green is there. Then you have the xanthophyll, which are yellow in color, and the carotenoids, which are yellow or yellow orange. Okay, so based upon these colors, okay, you have two types of spectrum, which is very important. Okay, so the two spectrums basically you have is a absorption spectrum and action spectrum. Now, what is this absorption and action spectrum? See, absorption spectrum means whenever you are drawing a graph, okay, certain changes will happen in that graph. So whenever you want to obtain an absorption spectrum, so the graph which is there, that will be plotted against the fraction of light which is absorbed by that particular pigment. Of light absorbed by pigment. Whereas action spectrum means what? Here you will see the rate of physiological activity which will be plotted against wavelength of light. Okay, so these particular pigments, or you can say the photosystems, pigments which are there, they are organized where in the thylakoid membrane, but it is having two photosystems, which we are going to see PS1 and PS2. Okay, so two different photosystems. PS1, PS2. So photosystem 1 and photosystem 2. Okay, so first one which we are going to see is the photosystem 1 or the light reaction also. See, now either of the one PS if you're going to take, okay, it'll be like having one specific chlorophyll. It's not that chlorophyll A, B, xanthophyll, carotenoids, everything will be present over there. Okay, so each photosystem is going to have a specific chlorophyll. Okay, so photosystems, they will have what? Specific chlorophyll, which is A. Okay, and plus other accessory pigments also will be there with some proteins. Fine. So whatever chlorophyll A is there, that forms the major central part for this photosystem, which what it does is it is going to take the sunlight. Like it acts basically as an antenna. Okay, so it will take all the sunlight and it will provide to the particular photosynthesis reaction. Okay, so if you consider the PS1 and PS2, so PS1 is P700, and here you have P680. That is the nanometer, means whatever light it is going to absorb, the wavelength of the light, how much it is for the PS1 it's 700, and for the PS2 it's what? 680. Okay, so the first reaction that we are going to see is the light reaction. It's also known as the photochemical phase. Now, this phase is a light reaction, so obviously it's dependent upon what? Light. Right? So here, whatever pigments are there, they're going to absorb the light energy, and in turn, what they're going to produce is the ATP, which is nothing but the energy molecule. So it absorbs light and produces ATP. Now here, this includes 4 steps. Okay, for the light reaction to happen, there are 4 steps which are included. So the first step is the light absorption. Second, splitting of water. Third, oxygen release. And fourth, formation of ATP and NADPH. Okay, so this is going to be later used in what? The biosynthetic phase. So actually what happens over here? So first thing, that is the, what do you say? The light absorption spectrum which is there. So here what happens is the pigments which are there, they get organized into 2 discrete LHC. Now what is this LHC? light harvesting complex. Okay, so we are going to see the first step. So in this, what happens is, the first thing is, whatever pigments are there, they are going to get organized into two discrete LHC. LHC means nothing but the light harvesting complex. So initially this is going to take the light from the sun. Okay. So now this LHC you will find in both PS1 also as well as in PS2. Fine. So now what is this LHC? LHC is nothing but, okay, it will have many, what do you say, pigment molecules. Okay, so it will contain all the pigments except a single chlorophyll A, okay, which we are going to see in each of the photosystems individually. So what happens over here is, this LHC, in this whatever pigments are present, okay, that is going to absorb the light according to the photosystem 1 and 2. For example, I told you 700 is for the 1 and 680 is for the 2, right? So this is how that LHC is going to work. Okay, so LHC is basically made up of what? Hundreds of pigment molecules. Okay, so the pigments which is in the photosystem 1 and 2, they are going to absorb the different wavelength of the light, right? So over here, you will be having a single chlorophyll A. The single chlorophyll A pigment, what it is going to do is, it is going to act as a reaction center. See, there are many different pigments I told you, A, B, and so on. But over here, whatever the LHC is there, it will have the hundreds of pigments except the chlorophyll A. And the chlorophyll A is separately going to act as what? A reaction center, right? So, reaction center means what? Each of them will be having a different wavelength, which is 700 nanometer. That is the highest peak for the PS1. And for the PS2, the highest peak is 680 nanometer. Okay, so this is in general about how the light reaction is going to take place. Now, one more important thing over here is what? What do you say, the electron transport system. Okay, see, obviously the electrons will be there, which is going to get transport, and it is going to what do you say, run this particular process, right? So one important thing that you have to keep in mind over here is, the chlorophyll A molecule is the reaction center for the photosystem 1 and photosystem 2, and other rest of the pigments or accessory pigments that will be present in this LHC. Fine. So now let's see the electron transport system. Now, basically, there is something which is called as what? Photophosphorylation. Okay, that we are going to see over here, the first one. So before that, let's go to the electron transport system. Electron transport system. What is this electron transport system? Now, already we know that the reaction center is present, right? So whatever the reaction center of the particular photosystem is there, it is going to absorb the light of a particular nanometer. And when it does so, it will cause the electron to become excited. And what happens when the electrons get excited? They are going to accept the electrons, and they are going to pass through this electron transport system, which is called as the cytochromes. Okay, so electron transport system means what? The reaction center of, take PS2 over here, it is going to absorb, what is a nanometer where it's going to absorb is the 680. So basically it comes under the red region. Okay, and because of this, what is going to happen? The electrons gets excited. And when the electrons get excited, they are picked up by an electron acceptor. Picked by whom? An electron acceptor. And then it is transported through this electron transport system, which is called as the cytochromes. Okay, so now what will happen is this electrons will be passed down to this electron transport system or the chain. Okay, then it will go to what? The PS1. So from the PS2, where that electrons are going to the PS1. Now in PS1 also, what is the nanometer of or the wavelength of the light is the 700 nanometer. So here also what is going to happen when it reaches the 700 nanometer? Here also the electron gets excited. And again that electron acceptor will accept that particular molecule, and then you will have a redox potential. Okay, so whenever these electrons are passing downwards, okay, this is called as the downhill direction. So when this is happening, what's going to happen is energy will be released. That is ATP. And this energy is going to reduce what? ADP, that is adenosine diphosphate, to ATP, and NADP, nicotinamide diphosphatase, to NADPH. So whatever this transfer of the electron is there, this whole system, that is from the starting from the PS2 of 680 wavelength to the 700 wavelength, the entire thing is called as what? Z scheme. Because the shape, when this happens, no, that shape is like in a Z form. So hence that's called as what? A Z scheme. Okay, so it's like, I'll just draw in short. Okay, suppose here it's the light. Okay, you have the electron acceptor. Here also one electron acceptor. This is for the PS1 and PS2. Okay, so take this as PS2 because it's the first one. PS1. So you have the LHC, light harvesting centers. So from there it will go to the first one. And then here to the electron transport chain. Electron transport chain, ETC. Okay, what will happen is it will go to the LHC of this particular PS1. And then again, the same thing is going to happen. So when this is happening, the ADP is converted to ATP and NADP is converted to NADPH. So this is a Z, what do you say, a shape that particular process is going to take place. So hence it's called as a Z scheme. And one more thing is during this, there one more small reaction occurs, which is called as what? Photolysis of water. Photolysis of water means what? Due to the sunlight, the water molecules break down. So this photolysis of water is going to release the electron. And in turn, to whom will it provide? To the PS2 for the further reaction. And also, because it's a photosynthesis process, so oxygen is also released. So this is only called as what? The electron transport system. Okay, so starting from the PS2, it will end in the PS1 at different manometers such as 680 and 700. And in between, there is going to be a photolysis of the water, so that what happens is the electron gets transferred from the PS2 to PS1, and there is a reduction of ADP to ATP and NDP to NADPH. And this all process takes place in Z scheme, so means Z direction or Z shape. So hence it's called as a Z scheme. Okay, so this also can be told as a cyclic and non-cyclic photophosphorylation. Cyclic or non-cyclic photophosphorylation. right? So in the cyclic, the first step will happen, and then sorry, in the non-cyclic, the first step will happen, and then the cyclic is going to this is for the PS1 and this is for the PS2, right? Cyclic and non-cyclic. Fine. And the important step over here is the splitting of the water molecule. That's it. So here you should know the differentiation or difference between the cyclic and non-cyclic photophosphorylation. So what is the difference between cyclic and non-cyclic? Okay, so first of all, in the cyclic, what happens is here photolysis will take place, but here it doesn't. Okay, then the second step, second difference is what? Here PS1, PS2 both will be there, but here only PS1 will be there. Here because it's a cyclic, so electrons are, what do you say? They are released, means they are no, they are cycled, you can say. Okay, they are cycled, and here they are not cycled. Okay, then the fourth one. Okay, sorry. Here one correction. Okay, here just a second. Mhm. Yeah. Okay, so in the cyclic, actually, here PS1 is involved. Okay, I just wrote the opposite. And here PS1 and PS2. And here no photolysis, and here photolysis. Yeah. So here in the cyclic, what happens is just the ATP is formed. But in the second phase, in the non-cyclic, what will happen? Both ATP and NADPH will be formed. NADPH. So here in the cyclic, what will happen is oxygen is not liberated. Whereas here it's liberated. So this is the difference between the electron transport system types, that is a cyclic and non-cyclic photophosphorylation. Yeah. Okay. So chemiosmotic hypothesis. Okay, that is for generally for what? The ATP formation. So this hypothesis was proposed by Mitchell in 1961. So what does this hypothesis actually state? It states that the ATP synthesis which is there, it is linked to what? It's linked to the development of proton gradient across the thylakoid membrane and mitochondria. Now see, now mitochondria is nothing but the powerhouse of the cell. So that's an important organelle. Whereas thylakoid is also important because it is going to help in the absorption of the sunlight. So here in this process, what is going to happen is we are going to see what is the development of the proton gradient across the membrane. Means the protons which are produced, how does it actually differ? Okay, when it's going through all this particular process, that is conversion of ATP to ADP and NADP to NADPH. Okay, so the first thing that happens over here is what? Splitting of water molecules, which we already saw, that is the photolysis of the water molecules. Now, this particular photolysis of water molecules takes place where? Inside the thylakoid, which is present in the chloroplast. Okay, so when the splitting happens, this H2O is the water molecule. So when the splitting happens of the water molecule, what is produced? Hydrogen ion. Now, what is this hydrogen ion? It is only proton, right? Now, we already know that the electrons which are there, it will pass through what? Two photosystems, that is PS2 and PS1. So, along with that, this proton, that is the H hydrogen ion, it is also going to